Hi everyone, in this video we're going to look into nucleic acids, which are biomolecules that carry and manage genetic information in cells. Now in very practical terms, nucleic acids are how biological systems can control genetic information, copy them, or use them in order to influence or control cellular activity. Now specifically referring to the structure of nucleic acids, they're basically just large polymers that are built from smaller repeating units called nucleotides, and their properties such as their stability, specifically referring to DNA, base pairing, hydrogen bonds between the nucleotides and sequence, that all kind of really makes heredity possible. Right. In terms of nucleic acids, there are mainly two types of nucleic acids that we have to know. First is DNA, also known as deoxyribonucleic acid, and second is RNA, known as ribonucleic acid. Now, DNA is primarily responsible for long-term storage of genetic information, and it's also a very stable sort of molecule, especially seen in comparison with RNA, which is a very versatile and volatile molecule in comparison to DNA. And because of its stability, molecule cells rely on DNA to preserve genetic information in sequences over time and across cell divisions. Next is RNA, and RNA is primarily responsible for using and transferring genetic information, and compared to DNA, it's much more dynamic because it can act as a messenger or even a functional molecule. And we also have hypothesis that RNA can act as a catalyst or an enzyme because of its reactivity, and hence the RNA world hypothesis. And so RNA is definitely much more flexible in terms of how it can be used. And although DNA and RNA are very much related chemically, it's very important to kind of distinguish them. And in our next slide, we will proceed to do so. Right, so in this slide, we're going to go over the functional and structural differences between RNA and DNA. First difference is to do with the sugar that's used in these respective nucleic acids. First of all, in RNA, we have ribose acting as the sugar. And in DNA, we have deoxyribose used as sugar. Now, the difference is that in RNA, in the ribose sugar, we basically have an OH hydroxyl molecule. We basically have the presence of an oxygen group. But in the same group, in the corresponding DNA, deoxyribose sugar, we have just the hydrogen group. So we don't have oxygen in DNA compared to RNA. And the presence and absence of oxygen is very important because the absence of oxygen in the OH group, basically leading to a hydrogen group in DNA, is very important because that contributes to DNA's overall stability, and that is why the job of the DNA is to store genetic information reliably. Second, to move on, is the difference regarding bases. We have uracil acting as the complementary base for adenine in RNA, but in DNA we use thymine instead. So in DNA, the adenine pairs up with thymine, whilst in RNA, uracil pairs up with adenine. Third difference is that RNA is just typically single-stranded, whereas just DNA is double-stranded and therefore forms a double helix. And the double-stranded structure just stabilizes our DNA structure and also makes it accurate for copying, possible through complementary pairing. Fourth difference is regarding the location of these respective nucleic acids. RNA can be found within the nucleus and even outside the nucleus because it's involved in processes like gene expression, and it can occur in multiple compartments. However, DNA is often just confined into the nucleus and in some organelles as well, and this is because its role of having to preserve that genetic information and store genetic information. Right. Third is replication capacity. Because we have the RNA worlds hypothesis, which is basically that RNA was a fundamentally initial rising molecule from which life began, we do have the hypothesis or the understanding that RNA can self-replicate because it has enzymatic properties, and this again has to relate to the presence of a hydroxyl group, which is very reactive, and so it can act as a catalyst, ribozyme to be more specific. However, for DNA, we need other molecules, other enzymes to assist its replication, so we generally have the understanding that it cannot self-replicate. Right, next we're going to move on to the nitrogenous bases that are involved. So nitrogenous bases is basically where all the sequence information is stored and encoded, and they're very important components of nucleotides because they vary for nucleotides and they also carry a lot of information. They generally are composed of elements carbon, hydrogen, oxygen, and nitrogen, and their specific structures allow them to form just specific selective hydrogen bonding sequences and patterns, which is why we have this sort of idea of base pairing being very much predictable rather than random per nucleotide that's involved in a nucleic acid. So we basically have 5 nitrogenous bases that are relevant here. We have adenine, also known as A, thymine T, uracil U, guanine G, and cytosine C. In terms of which nucleic acid uses what, DNA uses ATGC, and then RNA uses AUGC. So if you just see a uracil base pair, you can just immediately think of it as RNA, as opposed to when you see a thymine, you can definitely associate it with DNA. Right, in our next video, we'll be looking into the 2 types of nitrogenous bases, which is pyrimidines and purines, and then we're going to just look into their detailed properties in our next video. But that is it for today. Thank you for watching.